Hi, welcome to Bio121. This is Lecture 1, What is Science? Today, we will be covering what science is and how to do it. Our objectives for today, first off, be able to define biology, life, human, hypothesis, experiment, and control group. Be able to list and define 5 properties of life. Be able to relate humans to all other life on Earth and list steps in the scientific method. For those of you just joining us, this will be the things from which I will derive test questions and homework questions. So let's get going. What is biology? Biology is the study of life. The word biologos literally means life words in Greek. And biology is the study of all living things. Now we know that life requires certain chemistry, so there will be a chemistry focus to our biology class. Life also has defined characteristics, and we will define 5 characteristics of life in this lecture. And life shows something called homeostasis. So homeostasis is balance in life. It is maintaining a stable interior despite a changing exterior, which means that life is separate and different from non-living things. So, what's alive? Well, something to be alive, it has to have a distinct molecular composition. So that means it requires energy and matter. So thing number 1, requiring energy and matter. Thing number 2 is that it is made of cells. Things like viruses are not technically alive because they are not made of cells. All living things are made of cells. Third, all life maintains homeostasis. We will get into the how on this in later lectures, but for now, understand that homeostasis is a stable interior state despite changing external states. This does not mean that the state inside is always precisely the same, but rather means it is stable and counteracts anything that would try to imbalance it. Your temperature is generally around 37.5 degrees Celsius, sometimes a little higher, sometimes a little lower, but always coming back to around the same area. This is what homeostasis is, and we'll discuss this more later. A fourth thing is that living organisms respond to changes in the external environment. If it is cold outside, you will shiver to keep your body in homeostasis. So we'll see how responses are often maintaining homeostasis and in response to an external change. All living things grow and reproduce. This is thing number 5. Life grows and reproduces. Bacteria grow in size and then divide to reproduce. Humans grow in size and then reproduce, and we'll discuss the how later on. A last thing about what it means to be alive that we will not be covering much in this course, but we'll be touching on very near the end, is that populations of living things evolve and become more adapted to their environment. So, let's look at some examples. Is this alive or not? A warm black blanket. Well, it requires energy to stay warm. It has a distinct molecular composition, but it is not made of cells. A warm black blanket can transform energy from light energy to heat energy, but it cannot maintain homeostasis or respond to an external environment. Blankets don't grow or reproduce, and blankets cannot evolve. A population of blankets will not evolve. Next, a crystal. This has a distinct molecular composition. It does require energy to make more crystals, and it can grow and reproduce, but crystals are made of crystalline structures, not cells, and crystals cannot maintain homeostasis. They cannot respond to the external environment. Although crystals can bud off new crystals, they cannot evolve, and they will always look the same. What about a virus? Well, it does have a distinct molecular composition. It does require energy in order to survive, but it's not alive. It's not made of cells. It cannot maintain homeostasis. A virus on a countertop will get cold and will eventually dry up. A virus does respond to the external environment, especially when entering cells. A virus does grow and reproduce, but it requires a host to do so. And viruses can evolve, as we have seen with cold viruses and flu viruses. What about us? Well, we have a distinct molecular composition. We require energy. We are made of cells, and of course, a little joke, memes. We can maintain homeostasis. When we're cold, we're shiver. When we're hot, we sweat. We respond to the external environment in the ways I just mentioned. We do grow, and we can reproduce, and humans can evolve. So, we are alive. Well, let's talk more about humans. We are animals, and we are something called eukaryotes. So how do we relate to other things? Well, we are not the same as bacteria. We are eukaryotes, not bacteria. We are animals, not plants or fungi. We are in the chordates, not jellyfish. We are mammals, not fish. And we are in the order primates. The family we are in is called Hominidae. The genus is Homo, and the species is Sapiens. Often we would call ourselves Homo sapiens. So what does this mean? Well, being in the domain eukarya means our cells have a nucleus, and our cells have distinct things called organelles, which we'll be discussing in several lectures. Being in the kingdom Animalia means we have nervous tissue and muscular tissue. We will discuss this when we discuss tissues in general, and we'll discuss nervous and muscular systems in specific. We are in the phylum Chordata, which means we have a dorsal nerve cord. It's on our back. And when we're lying down, if we're lying on our stomach, it's on the top. This is different from insects, which have a nerve cord on their bottom side. We're in class Mammalia, which means we lactate and bear live young. And this will be the subject of our discussions on reproduction. We are in the order Primates. This means we have grasping digits, our hands and thumbs. We have binocular vision, our eyes are in the front of our skull. And we have extended care of offspring. We're in the family Hominidae. This means we have a palm and thumbs. We are bipedal. We walk on two limbs. And these thumbs are opposable and can touch all other four fingers. We are in the genus Homo, as defined as having a large brain, being able to use tools, and having complex language, so much so that you need to learn new terms, which we'll be doing in this class. We are in the species Sapiens. It means wise ones. And that's because we came up with a naming scheme, so we can name ourselves whatever we want. Hey, wise guys? So, let's look at the levels of organization of a human. First off, we look at the smallest things, the atoms and molecules. We'll be covering those first. We'll be looking at cells and cell structure then, tissues and tissue structures. These tissues make up organs. We'll be looking at organs in the context of organ systems. And this all makes up the organism. And that's about where human biology ends. Now, you will, in other courses, cover populations and social interactions, cover the community, which involves the other creatures around us, cover the ecosystem, which is the greater Washington area and where we live, and cover the biosphere, this blue marble that we live on. But those are four different classes. Here, we're going atoms through organisms. And how are we going to approach it? We're going to approach it scientifically. So, what's science? Science is a method of knowing things. Science is a way of determining what is a reliable method of knowing things. So we, as humans, tend to rely on anecdotes. What are our experiences? And how do our experiences shape us? If we eat bread and that bread makes us sick, we realize we should not eat bread. That's an anecdote, though. So this one time I ate bread and I got sick. That's an anecdote. That's not always going to be true for everyone. Other people eat bread and are fine. There is an approach called solipsism, which is becoming more common in our culture, and it's an approach of knowing things. What it says is that you can know yourself, and no one else can know you, and you cannot know anyone else. You'll hear it said, well, that's true for you, but not for me. That's solipsism, and we won't be doing that in class. We are going to do something called empiricism. Empiricism is taking experience and making it into experiments. So what is true for us can be true for others. What we can sense is probably real. And if it's not real, we can experiment to find out what is real and what is not. It's the idea that our lived experience and our experiments in it are reliable, and that if something is true for most people, it's probably true for you. So how do we approach this? Well, we use something called the scientific method. This is one of the things you will want to know for this class. We start off with observations. Hey, this bread made me sick. And we make a hypothesis, which is a tentative statement about the natural world. Something in this bread reacts with my body and makes me sick. Okay, we can use a hypothesis to make a prediction. If I eat any bread, then it will make me sick. And then we make a planned experiment. And this is where we have to move beyond the simple one-person experience. An experiment has to have a certain number of samples, a certain number of people who get a treatment, and a certain number of people who don't. And we will call these the experimental group and the control group. The experimental group will get something given to them. Say, give a bunch of random people bread, and give a bunch of other random people something that is not bread, and see if this makes everyone sick in the experimental group, or just see if everyone gets sick in general. If everyone gets sick in general, it's probably not the bread that's making them ill. So a good way of looking at this would be the drug trials. In this, you pick a large number of appropriate subjects, just general people in the population. You randomly divide the subjects into two groups. You treat the group equally in every way except one way. And that one way is what you believe will make the difference. In our aforementioned one, it was the bread. In this one, it might be a blood pressure medication. So you have a large number of people who all have blood pressure. You give some of them a drug that is supposed to lower their blood pressure. You give others a placebo, which would be a control group. This is just a pill that looks the same, and the people don't know if they got it or not. So the only thing that changes is going to be what the content of that pill is. Then you will take blood pressure measurements to actually measure if the blood pressure has gone down or not. And you'll compare the results. If the blood pressure is lower in the experimental group, then that supports the hypothesis that blood pressure medication is actually doing something. If they are equal, then that means the blood pressure medication is not really doing anything. If they're lower in the placebo, you might have a blood pressure medication that raises the blood pressure, and that would require further experimentation. We approach this with something called statistics as well, which we're not going to get into this in this class, but the class statistics is a good way of learning how to appropriately compare multiple groups. And over time, we use experiments to make something called a theory, which is greater than a hypothesis. It's more of a supported and well demonstrated idea or concept, like the theory of gravity, or atomic theory, or cell theory. In science, we need critical thinking. We need to think, what is the source we're getting this from? If you hear something such like, let's say, ginseng reduces blood pressure. Okay, what's the source? Ginsengisawesome.com might not be a good source. A scientific peer-reviewed published paper is definitely a better source. So are these people biased? Do they have the credentials to actually be in making these statements? This is critical thinking. What is the evidence? This one dude tried it, man, and his blood pressure totally went down. One dude is not sufficient evidence. A controlled experiment is sufficient evidence. So always asking yourself, is this source qualified? Is this source unbiased? Is a method of critical thinking we use in science. And we use statistics, which requires sample sizes, which requires control groups. And we believe in science that statistics are far greater than anecdotal evidence alone. So, how do we apply this to society in general? I know a lot of people in here are taking this course as part of social work. So how do we apply all this wonderful science stuff to, a real field like social work? Well, first off, science can predict things. We can use science to predict the weather. We can use science to predict when an eclipse will happen. We can actually use science to predict what strain of the influenza virus will be the most common next year and plan our vaccines accordingly. That is a use of science in society. Science can also predict what will happen if a population does not get enough vitamin C. They'll get scurvy. So this is a useful thing to improve the lives of people. Science, of course, can also improve technology and can improve medicine. It can also be used in less ethical ways, such as the overprescription of opioids, which we will look at later, and the impact that this had on society. Science and ethics is a deep dive that we're not going to take all the time, but we are occasionally going to look at the ethical implications of scientific things. For example, folic acid supplements were given to a group of women in a British study, and they found out that these folic acid supplements reduced birth defects. Well, that's great. That's an experimental group. But was it ethical to deny the control group something that could reduce birth defects? This is an ethical question we'll get to a little later on. And the fun thing about this course is you'll have time to discuss these ethical questions if you are attending the in-person lectures. So, with this in mind, let's get started on a wonderful course.